Good afternoon, everyone. I'm Jessica Kim Cohen, a reporter at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Expert Panel, Guideline-Driven Pharmacogenomic Testing and the Trajectory of Utilization, and it's sponsored by Agena Bioscience. Our panelists today are Chad Bousman, who's an associate professor in the Department of Medical Genetics, Psychiatry, Physiology, and Pharmacology and Community Health Sciences at the University of Calgary, and Vicki Pratt, Director of Scientific Affairs, Pharmacogenomics at Agena Bioscience. The discussion today will be moderated by Glenn Sawyer, Associate Director of Clinical Genetics at Agena Bioscience. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are several widgets to enhance your webinar experience. And with that, I'll turn it over to Glenn.
Thank you, Jessica. Thank you for the introduction, and thank you to GenomeWeb for the opportunity to co-host this webinar, which we hope the audience will find both educational and exciting. As Jessica mentioned, I'm Glenn Sawyer, the Associate Director of Clinical Genetics at Agena Bioscience. And it's our privilege to have two distinguished speakers with us today for this panel discussion. Jessica provided an overview, but I'd like to take a brief moment to highlight their numerous accomplishments in the field of pharmacogenomics. Our first panelist is Dr. Chad Bousman from the University of Calgary, where he leads the Psychiatric Pharmacogenomics Laboratory. His program spans PGX discovery and clinical implementation with a focus on improving prescribing for mental health conditions across life stages. Chad has published over 200 peer-reviewed papers and is a member of several societies, including the American College of Neuropsychopharmacology, CPIC, PharmVar, PGRN, and Stripe. And as if that wasn't enough already, in his spare time, Dr. Bousman is the founder and CEO of Sequence to Script, a software company assisting labs with translating PGX information into clinical action. Welcome, Chad. Dr. Vicki Pratt certainly needs no introduction to those in the PGX field, as her credentials are well established. She is the past president of AMP. She continues to serve with the CDC GetRM program for reference materials for molecular genetics. She's part of the National Academy of Medicine's Roundtable on Genomics and Precision Health, and the AMA's CPT Advisory Committee, just to name a few. We're extremely fortunate to have her as the Director of Scientific Affairs for PGX here at Agena. Welcome, Vicki. So the format of this webinar, we're going to have a roundtable discussion where we have some set questions that we'll ask our experts, and we'd love to get their opinion and their take on things developing in the field of pharmacogenomics. And our first question is that in recent years, there's been a lot of effort by leading societies to define which pharmacogenomic genes and variants should be included in standard PGX testing. With so many genes and so many variants to consider, it can be difficult for labs to decide what content should be in the testing that they offer. What resources should laboratories utilize to help them decide what to include in their PGX testing? And Vicki, I'd like to start with you for this one.
Sure, thanks, Glenn. First of all, I think at least when I was running my lab at Indiana University, one of the things I sort of looked at was what was really my patient population that we were going to test? Are we going to have a broad panel? Because that's going to impact the genes. Am I testing pediatric oncology patients? Am I testing advanced cancer patients with lots of polypharmacy? So really sort of defining that patient population was really important at first. And then I could go look at the resources. I mean, PharmG, and I'm sure Chad will agree, PharmGKB is a great resource, CPIC's a great resource, the FDA Drug Gene Association tables are a great resource, the Association for Molecular Pathology, the AMP tier 1 and tier 2 variants for the genes that have been reviewed already. So I think once you get that sort of patient population and the medications or the drugs that are applicable there, then you can start looking at the genes and some of the variants. Chad, what did I miss on that one?
Yeah, no, I think you pretty much covered everything. I would say that, PharmGKB is probably for me, it's usually the first stop. It's, it's kind of a one-stop shop. There's everything there. All those other resources that Vicki just mentioned can be accessed via PharmGKB. So I typically tell people to start there. It operates like a search engine, so it's really easy to use. You can go down rabbit holes on PharmGKB. That's the only downside. If you're someone that's susceptible to rabbit holes, you definitely will get down one at some point. And then PharmVar as well. I don't think you mentioned PharmVar, but PharmVar is also a good resource just to see what SNPs or what variants you need to test to make sure you're covering particular the star alleles that are listed in that AMP tier 1, tier 2, just to make sure that you're covering those appropriately. But otherwise, I agree 100% with what Vicki just said.
Thanks. And obviously, with the many that you mentioned, are the pluses or minuses with each of them? I mean, there's no, to my knowledge, I know everyone tends to go to the various sites. There's not one specific one-stop shop that gives you everything you need. Anything that strengths of one versus the other, depending on what people are looking for, whether, like Vicki said, she had mentioned, depending on your population, depending on the medications, anything you would suggest for any of those sites, one versus the other? Chad?
I mean, I do think PharmGKB is a good place to start. Because all of these resources, a lot of them have overlap. They have some of the same people on their advisory boards. They all work together. So I really don't think, if there is a limitation for one, another one picks it up, the slack. And the goal is not to have too much overlap between them. But that's what PharmGKB kind of brings everything together, so that you can just go to that one and then it branches off. I mean, all the tier 1, tier 2 alleles by AMP are on PharmGKB. PharmVar, you can link to PharmVar straight from PharmGKB. CPIC, Dutch, FDA, a number of other regulatory bodies, it's all curated there in PharmGKB. So yeah, like I said, there's a lot of information there. So I think I would
agree. Yeah, I'd agree.
Yeah.
I mean, the first stop I would be PharmGKB, because you can link out to the other spots.
Yeah, exactly. The challenge is this: once you're there, is figuring out where to go sometimes. And if you're not really sure, it takes some time to just get acclimated to the website and what it can offer. But once you figure it out, it's just a really good place to start.
Perfect. Thank you both. Appreciate the insight there. Then we'll move to the next question. I'll be on the screen here. So for question 2, the field of PGX is always advancing, and there's constantly new scientific evidence that's emerging, and it's supporting new or additional genes and variants. And it can be difficult for a laboratory to feel comfortable implementing a test or a panel for fear of not having all critical content included. Chad, I'll ask you first of how you would advise a laboratory to implement a PGX panel and future-proofing it as best as possible.
Yeah, this is a good question. I mean, I think the only way you can really future-proof is to test everything, a very comprehensive approach, and that's obviously not necessarily economically feasible at this point in time. When I think about this, though, for future-proofing, I think of there's 3 steps. Basically, what we were just talking about, being aware of the current and emerging pharmacogenetic evidence. If you don't know what the evidence is, then it's hard to future-proof. Understanding, and this is something that Vicki mentioned in the first one, understanding your end users, whether it be patients or clinicians. If you don't understand what you need, it's hard to future-proof as well, or what you may need. If you're a site that's not providing cancer care, then maybe you don't need to include cancer genes on your panel. If you might, in the future, start doing cancer care, then you might want to think about putting that on your panel. And then finally, selecting a platform, a genotyping platform that actually allows for flexibility. You can easily add, remove, change your assay as needed, hopefully with very little pain in the process. So I think most of us go with a modular approach. We start with, this is what we need today. Here's all the things we may need tomorrow. We're going to keep those assays there, and we'll add those as needed, or maybe we'll include those and we'll filter them out during the reporting process. That depends on local, decisions there. But those are the two main areas if you're not going to do a comprehensive approach, which I don't think many are able to do at this point.
Thanks, Chad. Vicki, any thoughts here as well?
No, and I totally agree with what Chad decided. When in my previous labs and previous life, when we set up a comprehensive panel, we were looking at a more global view of what we were going to do. And I think we went with the main commonly tested genes. If we think about 2C19, 2C9, 2D6, and some of the other ones, we really sort of went with those first. We went with more of a modular approach that we had this sort of core set that actually did pretty well overall with having a really good grounding of evidence around it. And then we added stuff in, or genes and variants in, as we had research projects and went through IRB, where we would report in and out as we got evidence or the evidence changed around some of the variants. So overall, I think if you look at the AMP Tier 1, Tier 2, and the genes that AMP's already reviewed, those were some of the more commonly tested genes that if you look on the NIH Genetic Test Registry or the College of American Pathologists reviews that they do of labs and what genes that they're testing. You can get a fair idea through some of those kind of resources.
Thanks, Vicki. And you had mentioned, obviously, with the AMP Tier 1, Tier 2, because there's so many different genes people are looking at, what's the typical, the level of evidence that we're looking for to qualify those different tiers? What backing scientific publications are typically considered when making those tiers?
So as Chad mentioned, PharmGKB, PharmVar, CPIC, and AMP, we actually all work together on that. So in general, we use the evidence levels that CPIC have assigned, or the CPIC work groups have assigned for the genes. When AMP does its reviews of the variants, our requirements, it has to have known function. It has to have a known impact on function. So it has to have a decreased function, a no function, or an increased function. Normal function variants aren't usually included. It has to have an appreciable minor allele frequency in a pan-ethnic population. It has to have a reference material or a positive control to ensure quality in testing. And it has to be testable or doable by the laboratory, because some variants are very, very complicated, like the hybrids, which isn't really always amenable to many laboratories, or some platforms for the laboratories. So we look at all that and sort of make our decision based on that. So the more common stuff's in Tier 1, as long as it has a positive control to ensure quality, and then the less common is usually in Tier 2.
Thank you, Vicki. And Chad, in your experience in your lab, do you find that coverage, looking at all the supporting evidence with AMP Tier 1, Tier 2, is that typically sufficient for the type of testing that you're doing in your laboratory?
Yeah, absolutely. I think, some labs because AMP is still making these for genes that is still outstanding. So I think in those cases, what we do is we apply we know the AMP criteria, as Vicki just said, so we apply it ourselves and hope we get close to what AMP's going to finally publish at some point, for that gene, because they're doing a much more rigorous approach potentially. But knowing what the criteria are, we can make pretty good guesses, I guess, at what alleles are going to be coming down the pipe. And then obviously if we're wrong, we adjust once they're published. But it's nice to have those criteria laid out. And I think everybody agrees on those are reasonable criteria. I think the one criteria and I think Vicki will agree with me on this, that's maybe the most frustrating is the reference materials. So we test for all tier 1 and tier 2, but some of the tier 2 alleles don't have reference materials. So that obviously that makes things difficult sometimes to determine if your assay is really detecting it correctly. So that's something for the whole field that, maybe expansion at some point of the GET-RM project, or, getting more samples with references. Some of these are quite rare, obviously, so it's difficult to do, but yeah, that's how we approach it at least.
Thank you, Chad. And I want to expand on that a little bit. Just Vicki, I obviously with the GET-RM project, can you speak on that a little bit? The goals there is because, as Chad mentioned, obviously we all know that reference material can be so difficult to obtain. So some thoughts on the GET-RM project.
Right, and so and have been working with the GET-RM project for many years, even before it was called the GET-RM. So with the folks there, Lisa Kalman, person there, Lisa Kalman, Dr. Lisa Kalman, who's at the CDC, we do work with the Coriell Cell Repository. We look at the collections that they have with the National Human Genome NHGRI. It's the NHGRI collection and other collections that Coriell has, looking for many of those variants. Some of those variants we can't find. We do reach out to different groups to see if they can submit samples if a variant is known, and get informed consent from individuals to try to enhance those collections. And it could be also that we need to work with our with vendors such as other vendors out there that do make reference materials and controls, and maybe if there isn't a Coriell cell line or a Coriell DNA, that's something that they can construct and make that available to the community.
Thank you. Yeah, I know it's always been a pain point for laboratories. So thank you for the details and some background on helping people to be able to get reference material. Now, the next question, third question, Vicki, we'll start with you on this one. In addition to the actual testing content, obviously there are differences from lab to lab into their approach and how they implement PGX. Can you comment on what you feel standardization of PGX implementation in the laboratory should consist of?
Oh, that's not an easy question.
Yeah.
So I'm going to go back to, again, if a lab's thinking about doing it, are you in a hospital system or working with like a research group? And do you have really looking at those patient populations? Is it a polypharmacy patient population where it's more like if you looked at the Kentucky Teachers Union population, that was more of a Medicare population where you'll see polypharmacy a little bit more than in sort of a younger population, or I should say below 65 population. Is it a cardiology? Are they cancer patients? Are you doing, are you working in some pain clinics where you're only thinking about pain meds? So I think when you're looking at standardization around that, that's a little bit harder question, because I think you've got to define and think about your patient populations more. I mean, we could say, oh, if you're going to do a broad panel, here's what you should think about as sort of the base genes, but everybody's going to have their flavor. Well, you know, I'm working with this and there's a lot of obesity here, and so they're taking these kind of medications. Or I'm working with cancer patients, so we've got a lot of antiemetics and antidepressants and pain meds and a whole other host of things. And so it's hard to say, what does standardization look like? I think you can have a sort of base, but then there's going to be flavors on that base depending on the patient population.
Thank you, Vicki. And Chad, in your experience with your laboratory and the successes you've seen, and I know you speak across Canada as far as PGX and implementation, any additional thoughts on what's worked for you or what you think about standardization of PGX implementation?
Yeah, I mean, I agree with Vicki. I think standardizing in your panel, a standardized panel, is going to be extremely difficult. What I spend most of my time in the standardization space, spend most of my time thinking about is the clinical interpretation and reporting standardization, because I do think we can make some waves there. So starting with even like the data schema and standards, if we're talking about integration into EHRs, there isn't really a PGX specific HL7 message. Like everyone has HL7 messages that are doing integration to EHR, but I've seen them, they're all different. Some similarities, but there's not a standardization there that would ensure interoperability across diverse EHRs. And I think we all agree that patients don't stay with the same provider or the same insurance, whatever. So they move. So their data has to move too. And if the standardization isn't there, that process is painful. And then there's just the reporting, those that we're still using PDF reports a lot here in Canada, having some standardization of not necessarily the look and feel of the report, but the content, what is in there, the nomenclature, the terminology used, the recommendations that are being provided. I mean, we looked head to head at a number of labs. We sent samples to the same lab or same person to like 5 labs, and then we just wanted to see, do we get the same results back? The genotypes were almost 99%. The genotypes they agreed on the genotype, but then when you looked at the actual reporting, like what do you do with that genotype? 60% agreed on what they should do with that genotype for the same drug. So there's obviously room for standardization on the reporting aspect. I think we still have a lot to do there. And I don't have a great answer of how to do that. It's just bringing more and more people to the table and hoping we can grab some consensus.
And again, I've seen, through the evolution of PGX, right, just that question, right, like on the reporting side, standardization, just so much information thrown at clinicians who are just trying to make a decision quickly and in the best interest of their patient, right? And so I've seen more of a move away from just the standard, red, yellow, green, to providing relevant details. Vicki, any thoughts on standardization of reporting from your side as well? I think Chad spoke to it very eloquently, but any additional thoughts?
No, I totally agree with what Chad said. But I think, and laboratory is somewhat ignored as the practice of medicine. So when you have laboratory directors who, I think if you go into any EHR and any medical system, that doctors don't present the data the same way. If you gave them like the same patient, they would do their write-ups slightly different. And I feel that laboratories have done that throughout, my entire career. You'll get societies or recommendations. You can see in CPIC, there's recommended language for CDS, but everybody tends to use their own little flavor on that sort of CDS or clinical decision support don't tend to use that same language, whether it's that sort of practice of medicine, they want to put their little flair or their sort of take on it compared to what the standard recommendation is.
Okay, thank you. Good insight. Appreciate you both. I know it's talking about both aspects of standardization, content testing, as well as the reporting. So appreciate it. Next question, fourth question we have, and Vicki, or excuse me, Chad, we'll start with you on this one. In recent years, we've seen signs of acceptance and recognition of the value of PGX testing with individual states and country coverage of pharmacogenomic markers, such as we've seen in California, Germany, others. What are your thoughts on what it will take to continue the momentum around the acceptance of PGX testing, and what efforts are needed to make it the standard of care?
Yeah, this is I think the $1 million question. I was just kind of looking at the Q&A too. I think this is going to cover some of what people are wanting to come here for as well. I mean, I look at this as, or look at this from working with and satisfying what I call the triad, which is the clinical practice guideline developers, regulators, and the payers. So all three, or at least one of those, you need to satisfy if you want to get paid, depending on which jurisdiction you're in and the current rules that are around that. You mentioned the California bill, that SB 496, I think, something like that. It says one of those three have to be present for it to be eligible. So ideally, we want all three on board. I personally think that the clinical practice guidelines is something we should really be focusing on. They're a tough nut to crack in a lot of fields. The cancer groups are trying to get DPYD, and most of us think that's a slam dunk, but it's still not part of clinical practice guidelines. You have clopidogrel and CYP2C19, same thing, and many other examples. I think once the clinical practice guidelines clearly state that this should be considered or even done before prescribing, it all kind of falls into place. Particularly the payers, I think, will look at that. Regulators are kind of on their own, depending on which regulator we're talking about. They're kind of on their own island, but certainly we can look at product labeling from many different regulators, and it's already there. There's definitely advice and to test. So I think it's bringing all those people together, maybe not all in the same room, but at some point working with them all to find out what is it that they need to make that happen. And then we have to, pick up our shovels and start doing it.
Thank you, Chad. Vicki, your thoughts as well?
Well, I mean, I think at least in the U.S., there's been the American Cancer Society Cancer Action Network has been working at including California in that as a state by state to include biomarkers. It really, I think, originally was around cancer, but the way the language is written, it includes pharmacogenetics as well as other biomarkers in there. So we're really seeing, at least on a state by state, more acceptance of biomarker testing, pharmacogenetic testing. Another thing that's more concerning here in the U.S. is there was a large commercial insurer that was covering pharmacogenetics testing, and just this year, late last year, they reversed their decision to cover pharmacogenetic panels, which I don't know that the evidence significantly changed. I don't know what the internal decision was to change around that. But it added a chilling effect for some laboratories who were working or getting paid by commercial insurers, and wondering if other commercial insurers were going to follow suit. On a more positive note, the Medicare in the U.S., the greater than 65 crowd, that program for covering panels has expanded throughout, especially with the MolDX program here in the U.S. And many states following the MolDX decisions for pharmacogenetic testing. And I do think, as Chad mentioned, it goes to practice guidelines, and they say they need more evidence. I don't know how much overwhelming evidence they need, or what that evidence is, but they say they need more evidence. I think that remains to be seen. Evidence is in the eye of the beholder, you know, where us that have already implemented pharmacogenetics think, oh my goodness, there's people dying from DPYD, there's overwhelming evidence. Why don't they see this? Where on the other side, they don't think that's enough evidence.
And along those lines, thank you, Vicki. It was a great perspective. And I know it's a topic of concern in the pharmacogenomics community, and I see even in some questions in the Q&A, particularly around DPYD and talking about, like, we feel like wow, there's so much evidence, it's a slam dunk, as Chad mentioned, yet it's not in guidelines. But I've seen some hope and progress, recent conferences attended where the dialogue is happening. What do you think needs to continue happening? I mean, just making sure that many of these societies that both of you are members of and the pharmacogenomics community continuing to have that conversation with those other societies that establish those guidelines. Chad, get your thoughts on that?
Yeah, I mean, I think the dialogue that's happening right now is great, and I think it just needs to increase. I think a lot of the dialogue has been, although maybe some of the people, the clinical practice guideline developers wouldn't admit it, but it's an education. It's a continuing medical education we're providing. And I think it is starting to sway them to where we think they should be. So I think some of it is ignorance. It's just because they're just not up to date on what the science actually says right now, the evidence says. And I'm not poking at them there. I mean, I don't expect everyone to know everything. So the dialogue serves that purpose. It's an educational opportunity for us as well to tell them, hey, look, did you know about this? Did you see this? And the things that I've been part of, I can see it happening in real time within the 2-day or 1-day workshops. The person that speaks first and then they speak last, and their views change in an 8-hour span. So I know it works. We just need to do more of that, I think, and I think it will come around. I was told once that clinical practice guidelines are typically 10 years behind the evidence. To me, that's unacceptable. And I think if we can make that at least maybe 1 or 2 years, that would be really a great achievement for us.
Thank you, Chad. Great perspective. And that's a good segue into the next question. And Vicki, I'd like to start with you on this question, because we've seen a lot of this in the recent years. But over the past year in particular, there's been an enormous amount of attention and awareness around artificial intelligence, around AI. Many labs are wondering how they can or should be utilizing AI to assist them with PGX testing. How do you see AI benefiting the PGX community, and where do you think it will be of the most benefit in the next 5 years?
I'm going to start with the next 5 years, because I really see, so your pharmacogenetic result is more or less, it's in your DNA. It's pretty static. And once you get that into the medical records are, they're fancy billing systems. So it's always the newest you'll see. You don't see the old stuff. I think AI is really going to benefit in clinical decision support, pulling out drug gene interactions that you might not see otherwise. I think I'm going to call it like a really fancy clinical decision support. I mean, the clinical decision support is still in its it's getting there. It's still in its infancy, but I think AI is really going to be able to enhance that and make that more point of care for the physician. So the physician doesn't have to look through a medical record. Well, what medications are they on? What was their PGX? What are the other drug gene interactions are going on here? I think that's where I really see it. I'm still waiting to see how well it'll work on pharmacogenetics. Is a lot of variance that creates the star alleles. Could AI help determine what the star alleles easier than some other algorithms out there, or novel combinations, and can it make a recommendation on whether that's a poor metabolizer or intermediate metabolizer, and to be able to pull that out faster. I haven't seen it so much there yet, but I think near term, this sort of point of care of drug gene interactions is really a place AI could probably shine. Chad knows way more about this than I could ever comment on.
Yeah, well, I don't want to set that expectation up too high. I'm certainly not an AI expert, but I do think in the next 5 years there are, in addition to what Vicki just said, there's some things that I think AI could probably assist with or be driven by. One is, we were talking about, I think the first question, the resources, and there's just so much knowledge base. Like, how do we? So I think AI, it's already doing ChatGPT. You can type questions in, and it spits out a summary that would take you several hours to come up to yourself. Now, of course, we all know that there's limitations to that, but in the next 5 years, I think there'll be improvements on the accuracy and less hallucination by AI machines, so that we can be confident in what it's providing us. At the point of care, I actually see it, something that could be done now is assisting or driving just-in-time education for physicians within the EHR. So we talk about chatbots, but this would be like chatbot on steroids. So actually giving some really good advice or consultation on the spot that you may not be able to get otherwise. Say you're working, a night shift on the weekend, and the consult service isn't available. Here comes AI in to save the day potentially. Now, I don't think we're there yet, but I do think in the next 5 years that might be possible. And then the final thing that I could see, and publications are already trickling out, are using AI in the physiological-based pharmacokinetic modeling, or PK/PD modeling, to try to predict or find associations in simulated environments that then we can then look for, test for in vivo. I think it's hard to recruit patients. So if we can simulate them, or virtual twins or virtual patients, and use AI to help us with that, I think it could expedite finding new markers and new variants. As we all know, we're very much in the PK space right now, right? And PD is extremely complex. And so I think AI could be really helpful with finding some PD variants or pathways for us to test for in the future.
Thank you both. I mean, that's great insight with AI. And another extension of AI that I want to propose to you is, like, attending a recent conference with a heavy focus on AI. One of the thoughts there was that, more immediate, again, over the next 5 years, is the potential for exactly what we were talking about on the last question, with alignment to clinical practice guidelines, right, and payers, and making sure that appropriate testing is being done for PGX, right? So, any thoughts on seeing alignment of how that's characterized with AI potentially?
Vicki, you want to go with that, or?
I don't know that I know the answer so much. I still sort of like Chad's example of, clinical decision support on steroids. Is there a way to, have that consult that pulls in, is it covered by insurance? Is it, is the patient's genotype? What's the medical practice? And pull that all in to make the physician a more informed, more informed management of that patient. The drug formulary, all that, pulling that all together to make it easier for a physician to manage a patient.
And I guess, I know it seems like a loaded question, and I guess what I was getting at is more of like what I've heard and what I've seen is there's so much talk and enthusiasm around AI, but there's also fear, right, of what it can be. And what I've seen in the community, what I've heard is that utilizing it as more as a comprehensive tool, exactly as Chad outlined, not as a replacement or supplement, but a tool to help those clinicians make a more informed decision.
Yeah, no, I do agree with that. Like, I do believe it's still going to require, that's why I like AI assisted instead of AI driven. I think we're way more than 5 years away from anything AI driven, in at least in the medical care space. But I do think it's already assisting in some regards, and I think it's just going to continue. So I think we have to lean into it, have a healthy skepticism, but we still need to lean into it. Resist it. You're going to go the way of the dinosaur or the dodo or whatever thing you want to pick, because it's happening. So I think lean into it and harness what it has, and let's use it.
I do see that there are opportunities, there are tests that are coming down the pike and already being offered that are using AI, but it still has to go through that sort of analytical validity, clinical validity, make sure it's spitting out the right answers and not spitting out wrong answers. I mean, it's still a patient we're taking care of at the end of the day, and we want the right answers and not the wrong answers.
No, thank you both. I know that was more of a philosophical with this last question, right? So Vicki, thank you for jumping on it first. Appreciate that. The last question that I want to pose to both of you before we open it up to Q&A, just any quick thoughts. Are there any other considerations or things related to PGX implementation that keep you up at night, or that we haven't already discussed, other than AI keeping me up at night? Any other things for implementation or PGX?
I guess the one thing that keeps me up at night, and this is why I've been leading the AMP recommendations that we've been publishing, is that for testing, for drug labels, to make sure it's pan-ethnic, a lot of the initial research studies were only done in white people, and the US and the world's made up of a lot more than white people, and to make sure that we're really people that are implementing that they aren't just testing only white people, that that we're thinking about a global society and variants, while they occur in more certain genetic ancestry, certain variants there, but to think more globally of that the world is diverse. That's the one that keeps me up at night, and that's what drives me for the AMP recommendations team that I'm leading.
Thank you, Vicki. Thank you for your efforts and all of that too. I know you're working hard at all of that. Chad, any last thoughts for you?
Yeah, I mean, I think I've mentioned most of the things that would keep me up. I mean, the one thing, and I wouldn't say that I'm trying to do my part in this, but I think it's a global thing, is more on the education piece. Both healthcare providers as well as patient education. I mean, it's a huge lift. We can't just do CMEs and workshops because the people that we need to reach don't attend those. So how do we educate the frontline general practitioners working 80 hours a week, have no time to log into a webinar? How do we get to them and tell them what pharmacogenetics can do for them and help their patients? And then also educating patients about, managing expectations around what pharmacogenetic testing can actually do at this point in time. That it's not a silver bullet. It certainly can help some, but sometimes it's indeterminate at this point for some patients. So I think setting that expectation is also good. So it's more about spread and scale of that educational approach, and how best do we do that. So I guess that kind of keeps me up at night trying to figure out how do we do that, and working with others to accomplish it.
No, appreciate it. Thank you both for your insights. And I know we're running towards the end of the webinar, so I'd like to turn it back over to Jessica to open up for the Q&A session. Thank you both.
Thanks, everyone. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. Also, we'd like to ask attendees to take a moment after the webinar is ended to take our exit survey to give us your feedback. So to get into the audience Q&A, the first question that we have is, an audience member was curious if you could comment on panel versus whole genome sequencing approaches for preemptive pharmacogenomics, and whether there are efforts of looking to implement WGS pharmacogenomics testing in healthcare settings.
I mean, I can speak to our local here. We're kind of exploring this. I know there's others doing this as well. As far as preemptive whole genome sequencing, no. But since I'm part of the medical genetics department here at the University of Calgary, and so there are whole genome sequencing is being done, typically in rare disease settings. But these kiddos or family members are also taking meds for other things sometimes. So we can repurpose that whole genome. We can extract, there's no need for them to now do a pharmacogenomics panel. The whole genome's there. So we're using it as like a secondary analysis or repurposing, and trying to provide basically more value for that whole genome sequence. It's already been paid for one purpose. Why not use it again? So that's one example of how I can see it being used, and that may be preemptive or reactive depending on the situation. But I don't know of too many that are ordering whole genome sequencing for the purpose of pharmacogenetics at this point in time, at least not being covered, reimbursed. That might be out of pocket for some people that want to do it. I don't know, Vicki, if you know of any other places that are doing it, but
no. As you've mentioned, I've seen that repurposing of either a whole genome or a whole exome, and the whole exome does have some issues with deep intronic variants, that it doesn't do very well, and it doesn't do hybrids very well in 2D6. So that repurposing I've seen, as well as the whole genome, and like in the newborn setting where you're getting some of that. But an upfront whole genome, I think the reimbursement isn't there, the justification isn't there, the reason isn't there for doing that kind of preemptive upfront testing.
And agreed, and I think it's going to be lab dependent, right? Like speaking with customers and laboratories, it depends on the needs, right? There's all the different considerations that labs have to think about, right? Coverage, cost, turnaround time, all of those. So again, I think it's a deeper discussion with each individual lab. But yeah, good question.
Yeah, and another question we have is, if a healthcare provider is using a test that includes the AMP tier 1 alleles, but then AMP adds additional tier 1 alleles, how should providers handle that?
I think that does become a struggle as we think about that. I don't know that the AMP's gonna make significant changes from only tier 1. If it's in a tier 2 may move to a tier 1 if there's a reference material. They're all discussed in the AMP paper. I don't know that there's going to be significant surprises if something moves around. I think, as with any testing evolves and changes over time, and it may require retesting for the patient down the line. We've seen it in Fragile X when they used to only do it with chromosomes, and then they used to only do it with PCR, then required southern blotting. So, I mean, testing evolves over time. Resolution on chromosomes have evolved over time. So sometimes it may require retesting as that knowledge and recommendations change.
I'll just add, I totally agree with what Vicki just said. In this hypothetical example, if a new tier 1 were to come up, to me, it would depend a little bit on the patient's current genotype. So if they were star 1, I'd be a little bit more likely to say, yeah, let's reorder it, because maybe you're not really a star 1, maybe you have this new one. But if you already have a diplotype that has a function that this new allele is not going to change it, even if you have it. Like if you're a poor metabolizer and this is a no function allele, well, you're already a poor metabolizer. Why should we retest you? So to me, I would on a case-by-case basis, potentially, in this particular hypothetical example. Now, if you're having a whole slew of new tier 1, then that kind of goes out maybe the window potentially. But I do think there is potential clinical scenarios where you wouldn't have to retest necessarily, depending on the patient's genotypes.
I also think it's depending on how the reporting is set up, right? Like if you have that, like for those partners like Sequence to Script, like Chad's company, right? If you have the information, if there's new recommendations that come out, if that can be easily, flexibly updated and within the report structure, right? And how that's actually reported out to the clinician. I think that's a key as well.
Yeah.
Definitely. And another question we had was, with new star alleles that have an unknown effect on the function of the enzyme, as those are being discovered, how should providers be considering those when determining metabolizer status?
I think that's more of a research. Yeah, I think that's more of a research question than a sort of a clinical implementation question. You know, when I was at the university, you know, we would look for maybe new star alleles or new alleles if that patient, we called them ultra responders. They either responded one way or another that didn't really match their PGX profile. Sort of what Chad's, you know, they seemed like they were a poor metabolizer, but they were star one for something. But you really have to do a lot of research in there to determine the function. You've got to do some in vitro studies or functional studies or some sort of other studies. And that to me is more on the research side, not on the clinical and the clinical implementation side. And there are groups that are involved in that. CPIC has groups that look at what's been published on them. You know, was there a patient? Were the patients involved? What were their functional studies when they're trying to make those functional information around star alleles and new star alleles, even existing star alleles, but new star alleles, definitely.
I mean, this is going back to the AI question. This is where AI could potentially help us with these modeling, right? It's like, okay, we have these new alleles. We don't have blood levels on these individuals that we found the new allele in. So now we have to try to estimate what it is. If you look at PharmVar, you see a ton of star alleles that say indeterminate or uncertain or unknown function. So from a clinical implementation perspective, we steer clear of those. We don't test them because if they come up, then we end up with a result that says indeterminate, which is the surefire way of making your clinicians upset with you is making them wait 7 days for the result and then telling them it's indeterminate. That's not a good way to build partnerships with your clinical end users. So that's the nice thing about AMP Tier 1 and Tier 2. You know, if you test for all those, they all have a function. So you're guaranteed that you're gonna be able to provide a result to them. If you put on these no function or unknown function, unknown or uncertain function alleles, you're really shooting yourself in the foot a little bit. It's more like Vicki said, they're great for research, but not for clinical implementation. You want to stay away from those, in my opinion.
Yeah, and we have had a couple of questions related to reimbursement and wondering what are your thoughts on what it will take for labs to get paid for pharmacogenomic testing, and if you have thoughts on what it will take for payers to consider or reconsider coverage of pharmacogenomic tests.
Yeah, so I think it's going to depend on the payer. Chad has the luxury of being in Canada, and the payer model. Europe has different payment models too, and I can't comment so much outside the U.S. because I'm not as familiar with them. You know, I think what's interesting is it depends on what you're doing. If you're probably doing a single gene, doing 2C19 for clopidogrel, there's well-established payment and reimbursement in both the commercial space as well as the Medicare space. If you're doing panels where I think that's where it's getting, maybe your indication is only for 2C19 clopidogrel, but you're reporting this large panel, well, then that's where it becomes more confusing, that why are you running a large panel when we only need one result? And I think that's where payers are pushing back some. The Medicare says if you're doing it around antidepressants, the patient has to, the physician has to document that the patient has failed therapy before you can consider doing a larger panel where it might help guide it. So I think it's going to really depend on the indication, whether it's a single drug gene pair, which most large commercial payers actually cover, compared to a more larger panel where they may not cover it. And some of that goes to a lot of fraud and abuse that have occurred in the past. So where insurers were covering it and there was fraud and abuse occurring, so now they're once bitten, twice shy, their insurers are less likely to cover some of that. Don't know that I answered the question.
I don't have a whole bunch more to add. I'll just say from the Canadian perspective, I mean, we really haven't figured this out here either, even though we have a different payer model. But the one thing I have found that's useful, it sounds very simple, but is just ask them, what do you need to make this decision? And then you either got to go find, generate it. Hopefully you only have to find it and not generate it. Like they said, there's for some of these C19 clopidogrel, DPYD, the evidence is pretty much overwhelming. So sometimes it just needs to be packaged in a way that they can understand it and make a decision. In other settings, you may actually have to generate it through some types of study design or looking at claims data. There's a number of different ways to do this, but really demonstrating that the clinical benefits outweigh the risk of testing. And of course, always the elephant in the room is the cost. Is it going to save us money, or at least be cost neutral? If you don't have those two answered, then you might as well walk out the door before you get there, because that's all they're going to ask about. Even if you have the best clinical data in the world, it's going to come down to cost when they talk about payers.
Thank you all. It looks like we are up against time for the end of the webinar. We'd like to thank Chad Bausman, Vicki Pratt, Glenn Sawyer, and our sponsor, Agena Bioscience, If we didn't have time to get to your question today, we will try to follow up with our experts. As a reminder for all of our attendees, please look out for the survey after you log out to provide your feedback. And if you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you all for joining us for this Precision Medicine online webinar.
Thank you, everyone.